tous et à toutes. Donc aujourd'hui, on a un webinaire de FR qui va être fait par Rita Rebollo de BF2I. Voilà, super Rita, qu'on remercie grandement. Est-ce que c'est bon, Laurence, tu as vu pour pouvoir l'enregistrer?
C'est en train d'enregistrer.
Ah, super. Bon, bah, c'est parfait. Je te laisse la parole, Rita. Merci beaucoup d'avoir accepté de faire ce webinaire.
L'audio ?
Oui. La première fois, ça n'a pas marché, j'entendais rien. Et puis après.
Vous entendez ?
Oui, là, on entend bien. Il faudrait couper vos micros, s'il vous plaît, pour qu'il n'y ait que celui de Rita d'ouvert.
Comme ça, vous entendrez bien mes reniflements.
Excusez-moi.
C'est tout bon?
Bon, tous les micros sont coupés.
Top. Alors, je suis désolée, je vais faire ça en English. C'est plus fastoche pour moi. So, thank you, Patricia, for inviting me. I'm very excited because this is the first time that I'm actually presenting this data. I'm just putting a timer because I think I'm gonna go way over the time, so you just cut me when needed. So I am a researcher at the BF2I laboratory and for biologie fonctionnelle insectes et interaction. And the overall goal of this lab is to understand the interactions between the insect hosts, which are often crop pests, and their symbiotic bacteria. So we do this in two main model systems. We study Cytophilus, so it's cereal grain weevils, and this we do in the team of Aziz Edi. Federica Calevro's group study aphids, and the idea is that there are main questions that are asked within these two groups, and then all the knowledge that is gathered within these groups feed a third group that is very novel. It's led by Pedro da Silva, and the idea is that Pedro will study the structure and the functions of the proteins that are found by these two other groups, and then potentially try to find biopesticides to fight all these crop pests. So I'm part of Aziz Edi team. So we study Cytophilus species, and Cytophilus is a major crop pest and is responsible for around 30% of loss of grains annually. And the reason why it does that is because the female Cytophilus actually lays its eggs within a cereal grain, and then the whole embryo, larval development, and metamorphosis happens within the grain. So you can already imagine how damaged this is for a cereal grain. And then when the adult is about 3 days old, it exits the grain and then, of course, starts eating again and starts laying eggs everywhere. So you can see how damaging an infestation by Cytophilus can be for several different cereal crops. So if you know a little bit about cereal grains, you know that they are actually quite an imbalanced diet. And so Cytophilus has an intracellular bacteria that is able to complement this unbalanced diet. So the intracellular bacteria is called Sodalis pyrantonis, and this bacteria is contained within specialized cells that we call the bacteriocytes. So it's not everywhere in the body of the little Cytophilus, of the little weevil. It's within this very specialized host cell. So what you can see here are two bacteriocytes. In red, you see the tubulin, and in green, you see a marker of DNA and RNA. So this is marking all the bacteria. So you can appreciate how the bacteriocyte is simply full of these bacteria. And here is something that I always show, and my lab is probably bored that I'm keeping showing this, but these are 3 bacteriocytes that we put it in culture, and I was just experimenting with it, and I just wanted to show that one of them is going to explode, so you can have an idea of all the bacteria that are in C. There's so much bacteria in it, and we like to call this bacteriocytes just bag of bacteria. So I think it's good. It's 2 images that are important to for you to appreciate how tight and crowded the relationship between this intracellular bacteria and the host is. And in order, of course, to understand, so the main question of Aziz teams, of Sim team, is to try to understand the molecular dialogue between this bacteria and the Cytophilus, the host. And of course, in order for us to do this, it's easier to have the genomes of these two partners available. So the bacterial genome was available a long time ago, but the Cytophilus genome was a very, 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 very long journey into acquiring. So this started potentially, I think now it's probably around 10 years ago, maybe 9, but, and I'm just the tip of the iceberg, huh? I just arrived at the BF2I lab 5 years ago. So my contribution to this project is very small compared to everyone who's been since the beginning. And so the whole idea was to get enough DNA and then to sequence the genome of Cytophilus oryzae. It should have been easy, but it wasn't. And what you can see here is that there are many different labs, parts of the consortium. And I just wanted to stress and pay attention to the 3 first authors who did this through a very long period of time. So I think it's important. Most of them were PhD at the time, except Nicolas, who arrived as a maître de conférence in BF2I lab. And then you have the corresponding authors. So what happened, right? Why was this so difficult? So the Nicolas Parizeau and Carlos Vargas had this massive amount of data. I think we had maybe 300x of coverage. We had short reads, so Illumina reads. We had longer reads, we had PacBio, we had Oxford Nanopore Technology. So these are all different sizes and chemistry of sequencing, which helps you to build a better genome. And despite all this, we weren't able to find a satisfying genome assembly. What does this mean? This means that when we wanted to study a gene, and then we would look at our assembly, we would find that the gene was fragmented between different parts of the genome. We couldn't see the gene in a perfect scaffold, okay? And so this lasted for at least 3 different assemblies. It was quite hard because every time we needed to annotate everything. And then Nicolas collaborated with Marie-France Sagot, and I think it was her PhD at the time, Alex Di Genova, and they built a software that allows all these different reads, chemistries and size of reads to be used together to create a better assembly. And they indeed succeeded, so we're very thankful for them, and especially thankful for this project to be finally over. And here's the assembly. So for all the stats nerds like Nicolas and I, you can appreciate all the metrics, all the quality metrics of the genome. So it's a 770 megabase genome, so it's quite large for insects, even though other Coleoptera, so here you can see other Coleoptera genomes, they have similar genome content, but you can see that others have smaller genomes. And what I think it's important here is that the genome was finally at a good quality, meaning that we could use this genome to do further analysis down the road. But this doesn't really explain why was it so difficult, right? So what's in this genome? So a little scoop, there's a lot of transposable elements. So transposable elements are DNA sequences that are able to jump from one location of the genome to another one, okay? So we can simplify this, but we can separate the transposable elements into 2 main groups. You have DNA transposons that are able to move, or as we call it, transpose, from one location of the genome to the other one by cutting themselves from the black DNA line and pasting themselves to the gray DNA line. And then we have RNA transposons, or retrotransposons. And these transposable elements here will transpose via an RNA molecule. So they will copy themselves through an RNA and paste themselves in another location of the genome. So how does this whole transposable element dynamics work? If we think, it's me trying to make a scheme about transposable element life cycle. But if you think about an active transposable element copy, you think that this copy is actually able to transpose, as I just showed you, and then to colonize a genome. So this means that you're going to have in the genome several different copies of that original copy. And we like to call all those different copies here a transposable element family. So transposable elements are very successful at colonizing the genomes, and they have been virtually shown in almost all genomes analyzed today. And you can also appreciate how, so the pie chart here shows in color the proportion of transposable elements in each of these genomes depicted here. We don't care about the classes for now, but what you can see is that the proportion of transposable element really varies, right? We have half of our genome that is composed of transposable elements, while, for instance, Drosophila here has 12 to 15%. Maize, and I think lungfish was the latest one, have like 85, 95% of transposable elements. Can you imagine? Almost the whole genome are transposable elements. A side note, this is Barbara McClintock. She's the researcher who discovered transposable elements and working in maize. So transposable elements, super successful. What else? Despite this very successful thing, we still have almost all species develop mechanisms that control these transposable elements. So the main silencing mechanisms that is able to keep transposable elements on check are epigenetic mechanisms, and we are going to talk about that a little bit later on. So through time, these transposable element copies can actually accumulate mutations. They can suffer recombinations because they have similar sequences of DNA, and you can have extinction of whole transposable element families. And this is interesting because I told you that we have half of our genome that is composed of transposable elements, but there's a tiny, tiny fraction of those transposable elements that are actually still able to transpose. Most of it are just fragments of old transposition events. So for all the transposable elements that are actually intact in your genome, but silenced through environmental changes, like biotic stresses, abiotic stresses, through disease, cancer is very well known, and through mutation accumulation, these transposable element copies can actually reactivate and recolonize the genome. Okay, so this was just for you to appreciate how the whole life cycle of transposable element is. But of course, there's something missing in the little genome representation here, is that there's a tiny fraction of the genomes that are genes, right? And which is what 80% of the researchers work on. And these genes here in the genome can actually be modulated. Their expression and their integrity structure can be changed by transposable elements. So those little fragments of transposable elements who are unable to transpose, they can still contain regulatory sequences that can regulate, promote, for instance, the expression of nearby genes. You can also have those silencing mechanisms that are targeting transposable elements that can spread, although very rarely, towards nearby gene. You can also have a transposable element that inserts into a gene, or two transposable elements that recombine. There are a myriad of ways that transposable elements can actually change a gene expression of or a genome architecture. And this is one of the reasons that today is very well accepted that transposable elements are a major player in evolution. And this is just a couple of examples. Like the mice coat color is due to a transposable element that is misregulated. This is how Barbara McClintock described the transposable element. So the color of the kernels is due to untransposable element movement. We have the color of a lot of flowers or the shape of tomatoes. And then we also have, this is just a schematic, we also have proteins that are derived from transposable elements that are today integrated into biological pathways. And one of them are called syncytins, and they are able to form tissue that is called the syncytiotrophoblasts that is necessary for mammalian development in the placenta. Okay, so I think you get why I wanted to study transposable elements and how I actually fell in love with them while I was still a PhD in Cristina Vieira's lab. So what happened like with our Cytophilus genome? Let's get back to the original question here. So we knew that they had a lot of transposable elements. So now we needed to detect all of them, and we need to be able to say what they were. So detect and annotate. And this is Clément Goubert, who was a PhD student also from Christina Vieira's lab and also Mathieu Boulestek's. And he started this collaboration with Aziz Edi lab and Christina back on his PhD. And he actually moved on to his first postdoc in the United States and his second postdoc to Canada, and we still hadn't finished that. So because of these time differences while we were working, we always worked very late at night. I am not gonna lie, several times with a glass of wine or a glass of beer. So this is how we actually properly annotate transposable elements. But it was a hard journey again. But we did it, and there is 72% of transposable elements in the Cytophilus genome, which is quite huge. And what you can see here is that you can see a representation of the genome of Cytophilus oryza. So these are not all the scaffolds, right? There were 2,000 or something. So these are the largest ones. And you can see in orange here, the coverage of exons. You can see there is very, very light. And then all of these here are the transposable elements. So you can appreciate how transposable elements are definitely everywhere in this genome. Every little thing that is not an exon is probably a transposable element. And you can see how the DNA elements, this red track here, is very, very abundant. If we compare this with other insects, and these are not all the insects where the transposable elements have been annotated, just a couple of them, you can see that indeed Cytophilus oryza has a higher amount of transposable element content, and most of these transposable elements are DNA elements, are the ones that cut and paste, right? You can also appreciate how, as usual, right, there's a lot of variation in transposable element proportion when we compare species, even if it's insect species, even if it's Coleoptera species. And there's also a lot of variation on the type of transposable element. And this is the same. It just means that there's really a very important relationship, an intimate relationship between the host genome and the types and quantities of transposable elements. So of course, with all this transposable element, here's one of the questions that we asked after annotating them and taking a break from the genome is, okay, so this genome is full of transposable elements, but are these transposable elements active? Like, in order for them to be active, they need to transcribe, produce the proteins, and then jump, right? So we attack the first step. Are they able to actually produce transcripts? Are they transcribed? So Mariana, who is a postdoc in our lab, did this. She went and dug for RNA sequencing data for a mosquito, Aedes albopictus. And this is because the proportion of Ts is quite high also in albopictus, also done by Clément Goubert and Mathieu. And then Drosophila melanogaster, which I told you is very well known model system, 12% of transposable elements, and especially it is accepted in the Drosophila world that Drosophila melanogaster has high activity of transposable elements. And then we have a little beast here, Cytophilus oryza, with the high amount of transposable elements. So when Mariana takes the RNA-seq reads, so an analysis of the transcription, okay? So she takes all the RNA-seq reads, she maps against the genome and against the transposable elements, and she simply counts the number of reads that map into a T. So this is a very raw analysis. And what you can see here is that in Cytophilus oryza, we have a higher rate of a higher proportion of reads from RNA-seq that are mapping against transposable elements, meaning that the transposable elements are not only abundant, but they are also being heavily transcribed. So what happens if we look at different tissues, right? Okay, those transposable elements are being transcribed, but are they being differentially regulated between tissues? So we did the same analysis here, but this time we compared midguts and ovaries, and we actually do a whole comparative analysis between them. And what you can see here is that every little circle is a transposable element family. And we are looking at normalized reads, so at the expression of this family in the guts and in the ovaries. Everything that is gray here, it means that it's the same between guts and ovaries. And you can see that there are a lot of transposable elements, families that are differentially expressed between the guts and the ovaries. You can also appreciate how there are many more transposable element families that are overexpressed in guts compared to ovaries. Probably meaning that in ovaries, right, where your germline is, you have a tighter control of those transposable elements. So this was really interesting because it made us think that Cytophilus not only had this genomic burden, right, 72% of Ts, but also have potentially this transcriptional burden. And that transcriptional burden was different between when we compared two different tissues. So the logical question that we asked after was, okay, no, how are these transposable elements regulated, right? How come there are so many transposable element families that are expressed, and how are they differently regulated between the tissues? So again, we looked at the model system Drosophila melanogaster because everything has been done there. And we know that in melanogaster and in other species, transposable elements are the target of small RNAs. So small RNAs are small RNA molecules that are able to target transcripts. It can be genes, it can be transposable elements or anything else. Those small RNAs can target these transcripts and they can either slice them or block translation. So this is how they work towards the transposable element. So in Drosophila melanogaster, there are two different classes of small RNAs. There are different classes because the way they are made, their biogenesis pathway is different. And at the end, their size, their structure is completely different. So in the germline, and this is a very simplified scheme, Marie, don't hate me. So in the germline, we have piRNAs that are going to control the transposable elements, while in the soma, it's the siRNAs that are going to control the transposable elements. Again, it's not really important what they are, just know that there are two different classes. It is very well known, at least in the past 5 years, that in other species than Drosophila, and especially outside Diptera, you can have piRNAs in the soma. Okay, people have shown this by simply sequencing the small RNAs in the soma. But there is a lot of variation. Within Coleoptera, you can have species that will have piRNAs only in the germline, like Drosophila, and species that will have piRNAs present both in the germline and in the soma. So with that massive amount of transposable element in Cytophilus, we wanted to see how were those transposable elements being regulated, especially between the germline and the soma, and if it was like Drosophila melanogaster, or if it was like other insects that have piRNAs both in the germline and in the soma. So what we did is that we took guts and ovaries, and we sequenced their small RNAs. And then this is just the distribution of the size of those small RNAs, because the siRNAs are 21 nucleotides, while the piRNAs range between 25 and 30, 24 and 29, it depends. So what we can see here in the gut is the read number, so this is just the number of small RNAs that we sequenced. We can see that we have abundant siRNAs, while we still have in guts some small RNA that look like piRNAs, right? In the ovaries, it's a little bit the opposite. We have more piRNA-like molecules than siRNA. So it would seem that, we have a little bit of piRNA in the gut, and we have a lot of piRNAs in the ovaries. So this was just looking at all small RNAs. So what we did after, and I forgot to say, this is work from Elisa, Inés, and Mariana, and what we did after is that we wanted to look at only the small RNAs that were targeting the transposable elements, okay? So I'm going to show a lot of graphs like this, completely copied and inspired by Marie Fablé. And what you can see here is that when it's blue, well, every circle again is a T family. When it's blue, it means that it's significantly different between ovaries and guts. And when it's pink, nah, it's not significant, it's the same. If everything that is above the line is overexpressed in ovaries, everything that is below the line is overexpressed in guts. So what do we see? If you look at piRNAs, we can see that they target preferentially transposable element families in the ovaries compared to gut, okay? Again, each little dot is a transposable element family. So this family here means that it has more piRNAs in the ovaries compared to the gut. Okay? And what about the siRNAs? The siRNAs, you can see that you're going to have a little bit more in guts compared to ovaries, right? So you would seem that piRNAs are more expressed in ovaries compared to guts, a little bit like Drosophila, right? But if we look at the expression within one tissue, right? Here we're comparing tissues. How about within one tissue? If I take gut and I look at the transposable elements that are being targeted by piRNAs or targeted by siRNAs, and I compare their expression, I can see there's still a lot of transposable elements that have more piRNA targeting them than they have siRNA. So even though there is more piRNA in the ovaries, there's still a lot of families that are preferentially targeted by piRNAs. I hope I'm not losing anyone. Please stop me when needed. And in the ovaries, you can see it's very clear. Most transposable element families are the target of piRNAs. So what does this mean? This means that yes, piRNAs are present in the germline and they are able to target transposable elements. And in the soma, it's not like Drosophila. We see both siRNA and we still see a little bit of piRNA. And here's where things are a bit different from what has been done in the other systems, is that we are lucky enough in Cytophilus oryza to be able to do functional analysis. So we didn't stop just by showing correlation. We actually went down and tried to show that these piRNAs are indeed controlling and regulating these transposable elements, and this Cy or Pi are regulating these transposable elements depending on the tissue. So this is a crazy experiment, and I think Elisa was my postdoc at the time. She hated me for it a little bit because she injected 440 animals with double-stranded RNA that was going to target different genes of the siRNA and the piRNA biogenesis pathway. So we were able to silence these genes, right? We are removing the key genes that are producing those molecules. So the idea is that those molecules won't be present anymore, right? And she did all this in a young adult, and we wait a little bit for the double-stranded RNA to work. And then we sequence all the small RNAs and all the RNA. And Agnes, of course, did all the libraries necessary for this project. I'm sorry, thank you very much. So I'm only going to show the results for 2 genes, but we actually made 3. So this is Argonaute-2. It's a protein that is associated with the production of siRNA. And what you can see here is that you can appreciate the expression of this gene when we knock it down. So you can see that is really low. The silencing works quite well and nothing changes for the others. The GFP is our negative control. Well, our control. It means that we're just injecting double-stranded RNA against GFP, and because there's no GFP in Cytophilus, nothing's supposed to happen. Okay, and then we have COE, which is the POE aubergine for the Drosophilists. And you can see the same thing, that when you try to silence, so this is part of the piRNA pathway. And when you knock it down, you can see there's a very good silencing of the gene. So what happens with the small RNAs? And this was very stressful because it was such a massive experiment that we were very scared that it didn't work, but it actually quite did. So same graph, transposable element family, blue, super different, pink, not very much. But the only difference here is that if it's above the line, it's going to be overexpressed in our mutants, in our RNAi deficient animals. And if it's below the line, it's going to be overexpressed in the controls. Okay, so here it is. If we knock down the piRNA machinery by targeting COE, what do we see if we sequence the small RNAs? This is looking at gut. So the piRNAs, they go down, right? Because you can see that they are overexpressed in the control. So there's no more piRNA, so experiment really worked. However, the siRNAs, as you can see here, they are overexpressed in our RNAi deficients. And so this means that, and this was curious and potentially unexpected, this means that there is a compensation, probably a compensation mechanism, where if you deplete the piRNAs, you actually increase the siRNAs. And what is the result of this in terms of expression of transposable elements? So again, this is the same thing. Each little circle is a transposable element family, but instead of looking at small RNAs, I'm looking at RNA-seq data, and I'm looking at reads mapping to TE families. And you can see that we have all these blues above the line. So this means that the transposable element families, all the blue ones, are being overexpressed. So that means that the piRNA machinery is functional in the gut and is controlling all these transposable element families. Things are prettier in the ovaries because I think there's less, there's more piRNA, as we saw before, abundance. So we see the same thing. We see depletion of piRNAs, we see increasing siRNAs, and we see loads of transposable elements families that are being overexpressed. So it worked quite well. How about the siRNA machinery? So this is knocking down AGO2. So results for gut, we see there's some weird stuff happening for piRNAs. We see there's a little bit more in the knockdowns. But for the siRNAs, what were we expecting? We were expecting all the siRNAs to be down-regulated, right? But it's not what we're seeing, and there's no differential expression of transposable element between our RNAi and our controls. This was a bit disappointing, but what I can tell you is that unfortunately, even though I've shown you that our knockdown was super efficient, when we did, so meaning that you have very little residual expression of the gene that we are targeting, we actually, when we do the RNA sequencing, instead of looking at the transposable element, we look at the genes, and then we look at the gene that we're trying to silence. We actually see libraries where the knockdown is really good. So we have like 2% of 2% of residual expression, but we also have other ones when we have 30%. Because I think knocking down the system that you're using to knocking down the system that you're using, I don't think it works. It's probably a loop where it's a matter of how much protein you have. So it's not always very efficient. So unfortunately, we can't conclude for the guts. For the ovaries, and I see that I almost have an hour. For the ovaries, we see that there is weirdly a depletion in piRNAs and an increase in siRNAs. So this is the opposite of what we were expecting. But despite these changes, which look very similar to this, right? We see no differences in the transposable element expression between our RNAi and our controls. So unfortunately for the siRNAs, I think it's very complicated to conclude if they are indeed controlling those transposable elements, which is really funny because that's, for us, that wasn't the main question. The main question was about the piRNAs, right? Because siRNAs can regulate transposable elements, but now we are in a situation where we very well demonstrated that piRNAs are able to control transposable element expression in both the germline and the soma, but the siRNAs remain to be further studied. Okay, so this is pretty much all I wanted to show about this whole regulatory thing. And I just want to go through one last a part of my talk, which is come back to the, main subject of the laboratory, which is the interaction between the insect and the intracellular bacteria, right? And one thing that I started thinking about when I arrived in the lab, again, I'm very naive towards symbiosis. I come from a transposable element field, so this was new to me. But when I got to the lab, one of the things that I started thinking and discussing with Aziz and the team is that there's potentially a trade-off between the fact that we have so much transposable element, right? And they're being expressed and you need to control them. There's more RNAs and etc. And then in a couple of cells in the bacteriocytes, you have all those bacteria that are, of course, also being controlled and regulated by the host. And there are a lot of papers that show that symbiont control could potentially be done by small RNAs. There are a lot of studies that show that transposable elements are labile to environmental changes as pathogen infection, bacterial infection, and etc. So I know it's a very out of there hypothesis or idea, but I really wanted to try to see if we were able to show an interconnectivity or a trade-off between transposable elements and those in the symbiont bacteria. And I think that Cytophilus is a great model to do this. And the reason why I say this is because, so I didn't tell you this, but the bacteria that is inside the cells is actually able to form organs, okay? Those organs are called bacterium. In the larvae, it's a little bean-shaped bacterium around the gut. And then, did I say adult? In the larvae? And then after metamorphosis, you have a lot of different, so this is the gut, and a lot of different bacteriums that are formed, and they contain those bacteriocytes. So what you need to know is that the Cytophilus animals are inside the grain, right? And they exit only at around day 3. So when they are inside the grain, so this is at day 1, the cuticle is not yet formed. And as we, humans, when you go out, you need to be well dressed. So in order to protect themselves and to prepare themselves to exiting the grain, Cytophilus builds up a cuticle, which is very well known in Coleoptera. And this cuticle needs aromatic amino acids that are actually provided by the intracellular bacteria. So there's this massive expansion. So this is gut bacterial load, okay? So the gut bacterial load increases a lot until it reaches a peak where we know that the cuticle is formed. And once the cuticle is formed, the bacteriocytes, okay, they suffer apoptosis and autophagy until around a 2-week-old Cytophilus doesn't have the bacteria anymore. It's quite a cool system, right? So for me, this is really cool because if you think about it, you can analyze the impact of maybe cells containing not a lot of bacteria, and then cells with a lot of bacteria, and then potentially cells that are dying with not a lot of bacteria. So this was a nice way of checking for transposable element variation in a constant system, at least in a constant genomic background, and see if you have any influence in the transposable element expression. And this is what we did. We sampled at day 1, day 3, day 7, and day 9, RNA of guts, and we did RNA sequencing, and we looked at genes, of course, but I'm not going to talk about genes, and transposable elements. So what you can see here is the number of transposable element families that are differentially expressed between all these ages. For instance, there are 345 transposable element families that are upregulated if you compare D1 to D3. And then there's 225 between D3 and D7, and there's very, very little between D7 and D9. And to be fair, between D7 and D9, nothing happens. There's no transposable element that changes. We know from other projects that there's no gene expression changes. So this is very, very similar. But the good differences, the good, if we can say that, the main differences between genes and also transposable elements happen between D1, D3, and D3, D7. So another a part of or characteristic of the Cytophilus model that I think it's really good to try to understand transposable element and intracellular bacteria trade-off is that in the lab we are able to have we're able to deplete Cytophilus from their bacteria. So this is a artificially made aposymbiotic weevil, so it has no bacteria whatsoever. Of course, it only lives well in the lab. If you put it outside, you'll die. But for us, it's really good. And this comparison between, sorry, there's a bee. This comparison between symbiotic and aposymbiotic has already led the group to find a lot of key genes that are associated in the regulation of the bacteria. So if we include this aposymbiotic to our studies, especially to our studies of transcriptional analysis of transposable elements, we may be able to find transposable elements that are symbiotic specific. And this is what we did. I mean, this is Mariana analyzing the data that Agnes did 3 days ago, okay? So this is very recent. I'm just gonna show a couple of examples. So here, what do you have? You have this transposable element here that is a DNA element, cut and paste. And what you can see here is the expression of these transposable element families at day 1, day 3, day 7, day 9 in the symbiotic weevil, and the same thing in the aposymbiotic weevil. So this is an example of a transposable element that has different expression between time points, but pretty much the same dynamics between symbiotic and aposymbiotic. We have cases where the transposable elements are only expressed in the symbiotic. We have examples where the transposable elements is only expressed in the aposymbiotic. And these are cases that we're really diving into to try to understand what they are, where they are located in the genome. These are cases where the transposable element expression follows the bacteria dynamic in the gut, suggesting that the more you have bacteria, the more you have transposable element expression, or there's something linked to both of them. And then when you look at the aposymbionts, there's nothing happening there. So we are trying to figure out what those transposable elements are. There's not that many, huh? And if they have anything that would be interesting for us to study further. And I will finally finish with some science fiction's idea, which is, well, the trade-off is already a science fiction. But this idea is that we have several different species that belong to the Cytophilus group. Cytophilus aureus is the one that we sequenced the genome, and I just analyzed all the transposable elements with the team and etc. But we also have Cytophilus zeamais, who has a very similar content on transposable element, a very similar ecology, and a very similar amount of bacteria. Then we have Cytophilus granarius, who only infests silos, and it has 2/3 of the transposable element content in the genome. And it also has very little bacteria compared to these two. And then we have linearis. And linearis is a tamarind weevil, so it doesn't live in grains whatsoever. And linearis has half the amount of transposable element from the others, and there's no endosymbiotic bacteria. So this is a very, I mean, it could be for many different reasons, right? But this is, again, those little steps in a step that allows you to formulate a hypothesis that tells you that potentially, yes, there is a trade-off between you being able to control bacteria inside your cell and you being able to control those genomic parasites. And there's, I mean, nothing has been shown by what we have done. There's a lot of stuff that need to be properly demonstrated, but I think that could be a nice science fiction project that, who knows, that might work. So I would like to finish by thanking everyone who helped or who gathered the data and the knowledge that I presented today. The whole genome project I had already shown the people who worked, so I would like to thank them again. And then the entire transposable element regulation, transposable element expression, and etc. This is all part of a great team of girls. So Elisa, Mariana, who were postdocs in the lab, really did amazing works. And then Agnès Vallée, who's probably has her hand in all of the slides that I've shown today. And of course, Nicolas for the constant bioinformatic help, and Aziz for all the discussions and for recruiting me. I also thank all the team from SimSim, of course, and BF2I. Of course, always part of my project, especially the transposable element ones. Clément Goubert, who despite getting crazy annotating transposable element is still on board doing stuff with Cytophilus, so that's really cool. I already said thank you to Marie for all the discussions and the graphs and the funding that allowed us to delve into this transposable element knowledge in Cytophilus. There you go. Sorry if it's too long.
Non, c'est perfect, merci. Je suis sûre qu'il va y avoir plein de questions. Merci beaucoup. En tout cas, il y a du monde.
Est-ce que je stop sharing?
Ouais, comme ça, on peut se voir.
Comme ça, on voit nos têtes.
Vous pouvez rallumer des caméras. Alors, il y a Fédé qui lève la main.
Non, désolée, je voulais féliciter Rita, comme ça, c'est fait. Bravo, Rita.
Je suis très contente de ne pas avoir éternué.
Désolée.
Merci, Fédé.
Merci.
Bravo Rita, puisque je suis Fédé. Très bon talk, bien résumé, tu as bien résumé les dernières recherches. Merci. Très clair. Bravo.
Merci à vous de m'accueillir à BF2I.
Alors, j'attends les questions, moi.
D'habitude, j'ouvre le bal, mais là, je ne vais pas poser les questions à Rita. Hein. Yvan, il y a sûrement une question.
C'est ça le pire, tu pourrais.
Je pourrais, bien sûr, je pourrais. Sauf que tu dis que c'est de la science-fiction, tu es très honnête, c'est vrai, mais il y a pas mal d'éléments quand même qui commencent à étayer ton hypothèse sur le trade-off entre le contrôle des éléments transposables et celui des endosymbiotes. Et il est vrai que ce système qui contient des insectes avec une importance symbiotique différente, voire même absence de symbiote, comme chez les linéaris, permettrait éventuellement d'aller plus loin là-dessus. Mais je salue ton honnêteté. C'est vrai que Rita, elle n'avance jamais les choses s'ils ne sont pas, disons, complètement démontrés.
On a une main qui s'est levée, c'est Natacha.
Pour aller dans cette...
Allez, Natacha, vas-y.
Pour aller dans cette direction, est-ce qu'il y a d'autres systèmes où justement on observe... Alors d'une, où ça a été étudié, parce que je ne suis pas sûre que ce soit trop étudié. Et si oui, si on observe des corrélations un peu similaires entre le taux de transposition et la présence d'endosymbiotes?
Non, c'est pour ça. Et c'est ce qu'on m'a dit quand j'ai déposé un projet. On m'a dit rien n'est connu. Parce que rien n'est connu. Donc, et c'est vrai que c'est compliqué parce que On en a discuté, c'est compliqué aussi de le faire en fait, parce que même si tu as un point I où tu trouves un truc, il faut vraiment essayer de trouver le système qui te permette ça, de suivre vraiment cette accumulation-là. Et donc avec Aziz, on avait cette idée d'essayer peut-être de trouver différentes espèces de cytophilus, d'aller un peu plus large, d'aller essayer même d'aller voir les trucs qui sont dans le musée d'histoire naturelle, tu vois, et d'essayer là de faire une espèce de séquençage évolutif des éléments transposables, tu vois, pour voir effectivement, histoire de reconstruire l'histoire. Mais c'est vrai que il y a rien de connu. Pas directement, du moins, entre symbiote et taux de transposition. Il y a des choses connues avec bactéries, un élément transposable, tu vois, immunité, bactéries, mais spécifique là, non.
Et tu penses que du coup, il y a une différence entre les symbiotes intracellulaires, genre transmis verticalement, et des symbiotes qui sont transmis horizontalement, ou dans des conditions différentes, que cette corrélation elle pourrait être différente suivant le type de transmission, parce que du coup, tu n'as pas forcément la même interaction générale. Et par exemple, tous les
Ouais.
systèmes de Martin
Ouais.
ou de Fukatsu, où les symbiotes sont acquis pseudo-verticalement, mais du coup, il y a quand même un petit peu moins de... Ils sont pas là tout le temps, ils sont pas dans les cellules. Peut-être ça pourrait être sympa de regarder dans ces systèmes-là pour voir si tu trouves le même pattern ou pas.
Ouais. Ouais, et surtout que je pense qu'il y a deux choses. Il va y avoir le lien entre taux d'éléments transposables et bactéries, et puis il va y avoir aussi le lien entre potentiellement transcription qui peut être différent. Et dans le taux, on est obligé d'avoir des transpositions dans dans la lignée germinale. Tu vois, il y a des choses qui sont quand même intrinsèques, donc tu es obligé d'avoir des infections assez tôt pour
Ouais, c'est
pouvoir
pour ça que
avoir
je
un
pense
effet
qu'il
total.
peut y avoir une différence entre ces différents types de
Tout à
symbiotes
fait.
et ça pourrait être cool de regarder. Bon, en tout cas, c'était super. Merci.
Très bonne remarque, Natacha. Je me demandais si Wolbachia, pareil, bon, il fait les deux, elle fait les deux. Effectivement, ça pourrait être dans un deuxième temps, sachant qu'on a aussi des souches qui ont des Wolbachia en même temps que le symbiote nutritionnel. Et là, c'est vrai que c'est une marche de plus. Donc, il faut commencer par un système facile à deux partenaires. Et effectivement, des questions comme ça pour tester.
Et
Si,
un temps évolutif long, quoi.
voilà,
C'est surtout
ah,
ça, tu vois. Il faut que tu aies
ah,
un temps de coévolution entre les deux qui soit assez long pour que
absolument.
tu puisses voir un impact, je
Tout à
pense.
fait. Mais justement, ces contraintes, les contraintes évolutives symbiotiques peuvent être sûrement différentes entre des symbioses à transmission verticale stricte et des symbioses à transmission verticale et horizontale en même temps. Il y a une main de Yvan.
Oui, je vois, effectivement. Et puis, j'étais en train de regarder, il y a
Non, mais
Sylvie qui met un truc dans le chat. Alors,
On peut le dire par oral aussi, hein.
vas-y.
Non, c'est juste pour dire, on a travaillé avec. Merci, un super exposé, vraiment, vraiment chouette, hein. Nous qui ne sommes pas du tout des spécialistes, mais on vient de montrer avec Clément que les transposons jouent aussi un rôle dans le parfum des roses. Et voilà,
Ouais.
si tu peux l'appliquer à tes exemples, il y a la couleur des fleurs, mais il y a aussi le parfum.
Il m'a montré, c'était un gypsy, c'est ça aussi, non? Parce que, bon, on a tendance à discuter le soir, malheureusement, avec Clément, au lieu de profiter de notre vie. Et il me montrait les graphes assez sympas qu'il était en train de préparer et tout. C'était en révision, je me souviens. Et là, il a été accepté, c'est ça?
Oui, 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 là, il est paru, oui, oui, dans
Super,
les vols il y a un mois ou deux. Ouais, ouais.
super, bravo.
Voilà, ce serait bien si on en discutait, on pourrait en discuter à part, parce que nous, on n'est pas des spécialistes, et c'est vrai que ça pourrait être une occasion de faire des choses ensemble.
en
C'est
fait,
sûr.
on comprend. En fait, on comprend pas du tout. C'est un bout de copia. On comprend pas du tout
Copia,
le
c'est
rôle qu'il
Copia,
peut
c'est ça.
avoir. On comprend pas du tout le rôle qu'il peut avoir. Il y a pas de site prévisible de facteur de transcription. Il y a pas. Il est tout petit.
C'était pas un Copia bizarre, en plus, c'était un fragment de Copia, un truc comme ça?
Ouais, 38 paires de bases.
Ouais, ouais. Et je
On
m'en
comprend
souviens
pas
qu'il
son
savait pas
rôle,
trop.
quoi.
Ouais.
On
Bah ouais, on peut en
ne veut
discuter,
pas
c'est sûr.
Ouais, non, non,
monopoliser.
mais bon, c'est pas.
On veut pas monopoliser, on peut envoyer le papier, tout simplement.
Vas-y, Yvan, on t'a coupé la parole, du coup.
Oui, mais j'avais pas mon micro, donc ça tombait bien. Alors, oui, Rita, dans votre petite analyse évolutive que tu évoquais à la fin, en fait, avec les 4 espèces de cytophilus, donc tu as un gradient de pourcentage de TE dans le génome. Est-ce que vous avez une idée de l'âge, de dire que quand est-ce que ça a commencé d'une part à envahir et d'autre part, alors ça sûrement pas avec juste 4FS, après comment ça a commencé à être exprimé. Alors ça c'est
Ouais.
une question. Et alors, après la deuxième question, dans votre système, comme vous avez maintenant comment tu les appelles, les pi-RNA qui sont manipulables quelque part, puisque donc est-ce que vous pouvez avoir des approches un peu par génétique directe, mais c'est-à-dire que voilà, et chercher des phénotypes
Mm.
entre les plus et les moins de
Tout à fait.
l'expression voilà, des transposons via la manipulation des pi.
Tout à fait, c'est très intéressant. Je vais te répondre par la deuxième d'abord. Ouais, c'est super intéressant. Je collabore sur un très joli projet de Christina sur ça, sur les drosos, avec, j'espère, une future thésarde. Et c'est vraiment cette idée-là de se dire, si tu dérégules les éléments transposables et tu es capable de créer des lignées qui ont différents taux de transposition, est-ce que tu as des phénotypes différents? Et on pourrait effectivement faire ça chez Cytophilus. Le gros problème, c'est que ce que j'ai pas montré, c'est que le RNAi, pour l'instant, ce que je vous ai montré, surtout le RNAi de pi, c'est létal. Donc, en gros, on a fait bien sûr des analyses de survie que je vous ai pas montrées. Mais quand on fait ce RNAi, donc quand on essaye de éteindre le gène à partir de la larve, c'est le plus tôt pour l'instant qu'on peut aller. Ils crèvent tous à peu près à la métamorphose parce que le gène SIWI, qui est le gène qu'on a choisi, est un gène qui joue d'autres rôles que la régulation des éléments transposables. Donc, il faudrait qu'on trouve un autre gène. Sur la droso, il y en a, donc c'est un peu de temps. Et il faudrait qu'on trouve quelque chose qui soit un peu plus spécifique. Et le RNAi, je pense qu'il pourrait même être utilisé effectivement pour faire du long terme. Mais bien sûr, le kiff du kiff serait si on arrive à faire du CRISPR chez Cytophilus. C'est un biotransposteur. Donc, on verra bien avec le temps comment ça va se passer. Mais c'est une très bonne idée. La première question que tu avais par rapport à ce gradient et à l'âge des éléments transposables, Clément a fait une étude évolutive comme on peut, hein. On n'a pas de horloge moléculaire, c'est comme ça qu'on dit, on n'a
Ouais.
pas d'horloge moléculaire. Donc c'est comme on peut. Mais on a vu que chez Linearis, il y a surtout des vieux éléments line qui sont très très intéressants. Pendant que chez les trois autres espèces, on a surtout des vieux DNA et on a aujourd'hui, le plus récent, plein de LTR, comme d'habitude d'ailleurs. Donc je n'ai pas raconté dans mon histoire, mais les familles d'éléments transposables qui sont régulées par les pi sont surtout des LTR, parce que c'est les familles les plus récentes, mais elles ne constituent que 2%, 3% du génome de Cytophilus. Donc il y a encore plein de petites choses qu'on doit répondre. Donc le paysage évolutif de ces quatre espèces au niveau des éléments transposables est très différent. Et on aimerait pouvoir créer une horloge moléculaire pour essayer de dater un petit peu mieux ces différents événements de burst de transposition. Peut-être essayer de voir si avec des remplacements symbiotiques qui ont pu avoir lieu, si avec l'histoire écologique des cytophilus, on peut effectivement associer, hein, c'est que de l'association, de la corrélation, des dynamiques évolutives différentes des éléments transposables.
Rita, et les LTR, c'est eux qui sont les plus exprimés au niveau.
Ouais. Après, tu sais, il y a des petites familles d'éléments transposables hyper exprimés, mais on pense que ce sont des gènes. Enfin, je corrige. On pense que ce sont des éléments qui ont été exaptés, c'est-à-dire qu'ils ont été, qu'aujourd'hui ils ont une fonction dans un rôle, ils ont un rôle biologique dans l'autre. Il y en a un qui est très exprimé chez le symbiotique, dont je serais intéressée d'étudier plus tard. Il y a trop de trucs à faire.
Alors, est-ce qu'il y a d'autres questions? Non, on dirait que c'est bon. Bah, merci beaucoup, Rita. Moi, j'ai appris plein de choses hein, sur les CRNA, les miRNA. À chaque fois, je me mélange avec ça. Il faut me trouver un moyen technique pour retenir ça.
Non, mais moi c'est pareil, hein. Moi j'appelle Marie, hein. Marie.
Merci beaucoup en tout cas pour
Merci.
ces
C'est
super
Bien.
c'est une
exposés,
grande
très
famille
clair.
maintenant.
Merci beaucoup.
L'AFR est une grande famille. Il se
Oui,
passe des choses.
ouais, c'est bien, c'est chouette.
Bah, surtout, je suis un peu inbred quand même.
Et ben à bientôt.
Allez, au revoir.
Merci
Au
à
revoir, au revoir à tout le monde.
Au revoir tout le
Bon
monde.
week-end.
Au revoir.
Au revoir.